In this video, we're going to talk about the cell membrane.
So the cell membrane has four main components: a phospholipid bilayer, cholesterol, proteins, and something called glycocalyx. And so we're going to talk about what do each of those four components do. And if you recall back to our lecture on macromolecules, we talked about phospholipids. And remember that phospholipids are amphipathic, meaning that they have both a hydrophilic portion and a hydrophobic portion. And if you remember, hydro is referring to water. And if you think of a phobia is a fear, right? If I'm arachnophobic, I'm afraid of spiders.
The hydrophilic heads are water loving.
And for those are going to face both inside the cell and outside the cell where water is present. The middle part of the membrane are made up of these fatty acid tails, and the fatty acid tails, remember, are hydrophobic. They're water-fearing. And so they're actually shielded from the water that's both inside the cell and outside the cell. And so the main component of the membrane is going to be this phospholipid bilayer. And this is really important because the membrane is a very fluid structure. And so the phospholipids themselves also help to maintain the correct fluidity. Another component for the fluidity of the membrane is the cholesterol. And when we think of cholesterol, we typically have a negative connotation in our head, right? You think of, oh, I need to make sure my cholesterol is not too high. But in fact, your cells actually require cholesterol in order to function. And so cholesterol, in some instances, is actually a good thing. And the cholesterol in the membrane is there to basically keep the correct fluidity, meaning that it prevents the membrane from packing in too tightly and making a very solid structure. And it also prevents the membrane from becoming too fluid, in which it couldn't regulate what goes into or out of the cell. And so cholesterol is very important in keeping the cell membrane the correct fluidity. The next component are going to be the proteins. And for the proteins, we're going to talk about the functions in a minute. Proteins have a variety of functions in a cell. If you think of an erythrocyte, which is red blood cells, erythrocytes have about 50 different types of proteins embedded in the membrane. Some of these proteins are what we call integral proteins, meaning that they're actually embedded in the membrane. And some of these proteins are going to be peripheral proteins, which are just associated with the membrane. The next and last component is going to be the glycocalyx. And these are basically going to be sugars that attach to proteins or phospholipids, and they serve as binding sites and as cell lubrication and adhesion molecules. They basically are there to help also with cell-cell recognition, meaning that two cells can recognize one another. So we're going to talk for a minute about what do those proteins do in the membrane. And remember that I said that if we look at an erythrocyte, which is a red blood cell has 50 different types of proteins. And so you can probably imagine that if it has that many different types of proteins, that they probably do very diverse things in the cell. And so one of the things that proteins do in the membrane is for transport. These are going to allow substances to flow through these hydrophilic channels. Some things can cross the membrane on their own to get into and out of the cell. Others need a little bit of help. And so that help is through these proteins. And we're going to talk about these more later in the lecture. Another function is going to be for enzymatic activity. And these basically are enzymes that are going to speed up chemical reactions. They help chemical reactions to go faster. And again, at the end of this lecture, we're going to spend a lot of time talking about what enzymes are and how they function. Another really important component of proteins in the membrane is that some of these proteins act as what are called signal transduction pathways. And basically what that means is that these receptors have what's called a ligand. And a ligand is a signaling molecule. And the ligand and the receptors are very, very specific. And when a ligand comes in and it signals and it binds to the receptor, that initiates some sort of signal transduction pathway inside the cell that tells the cell what to do. For example, if you got a cut and you had a wound, you probably know that that wound is not going to stay there forever. Eventually that wound is going to heal. And one of the things that's going to happen is the cells in the area of the wound are going to send growth factors to the cells in the area, telling them to divide to repair the damaged tissue. And so these growth factors get sent, and the growth factors themselves are going to be the ligand, and cells have receptors for those growth factors, and that then sends a signal to tell that cell to divide. And so these are called signal transduction pathways. They're basically a way to relay a signal to allow cells to communicate with each other. Another important function of a membrane is cell-cell recognition. And this basically is going to serve as identification tags recognized by other cells. So if you remember in our cell lecture where we looked at the video of the white blood cell chasing the bacteria, you'll remember that the white blood cell didn't engulf the red blood cells. It recognized that those cells were self and that not to engulf its own red blood cells. However, it did recognize a signaling molecule on the bacteria, and when it came to the bacteria, you'll remember that it engulfed and it took in that bacteria. And so there are these proteins, typically glycoproteins, that help with cell-cell recognition. The next is going to be for intercellular joining, so allowing two cells to join together and kind of holding them together. And then lastly, attachment to the cytoskeleton and what's called the extracellular matrix. And this is basically there to help maintain cell shape and to coordinate changes to the cell shape. And so this is going to be important for maintaining cell shape. And so we're going to talk about now traffic across the membrane. How do we get molecules into or out of the cell? And traffic across the membrane is essential. You have to be able to get things into and out of the cell in order for the cell to survive. And if you think about it, for example, think of your own cells. If you don't eat, your cells can't survive. Your cells need to be able to take in molecules for energy. We're not plants. We can't make our own energy. And so the way that we get our energy is by eating food, right? And so our cells need to be able to take in those sugars and take in those amino acids and take in all the other essential nutrients that our cell needs to survive. And so obviously then cells need to be able to take in different molecules for energy. They also need to be able to get rid of metabolic waste products, so things that the cell no longer needs. If you think of breathing, for example, right? You breathe in oxygen and you exhale carbon dioxide. And you're going to see later that the reason for this gas exchange is for cellular respiration. And carbon dioxide is going to be a waste product of cellular respiration, and the cells need to be able to get rid of it, and the cells will have the carbon dioxide leave. It'll go through the bloodstream to the lungs, and you exhale it out. Additionally, cells need to be able to take in and expel many important ions. Sodium and potassium, for example. So if you look at this image here are these sodium and potassium channels. And neurons, which are the nervous system cells that communicate with one another, the way that neurons fire is simply through a change in the distribution of ions. And the way that these ions move is through these sodium or potassium channels. And all an action potential or a neuron firing really is a change in voltage, which is a result of a change in the ion distribution across the cell membrane. And so being able to move these ions is essential. If we think about calcium, right? You probably have been told when you were younger that you needed to drink, let's say, milk for calcium for your bones. So cells need to have calcium. Chloride. Chloride is an ion, and chloride we'll talk about later. A disease that results from defective chloride from defective chloride channels is going to be cystic fibrosis. And cystic fibrosis causes these defective chloride channels. And we'll talk about later about how that affects the movement of water across the membrane. So when we look at the phospholipid bilayer, remember that the bilayer is made up primarily of phospholipids. And again, phospholipids are amphipathic. meaning they have these hydrophilic heads and these hydrophobic nonpolar tails. And so the way that the membrane orients itself, again, is that the heads, which are hydrophilic, want to interact with the water both outside the cell and inside the cell. And the tails that are hydrophobic want to be shielded from the water, and they're going to be in the middle. So when we look at this membrane, again, there's a part that's hydrophilic and there's a portion that's hydrophobic. And this is going to basically have an effect on what types of things can cross or do not cross the cell membrane. So the things that can cross the membrane on their own are going to be things that are hydrophobic. So hydrophobic and hydrophobic, oil and oil, can interact with one another. And so these hydrophobic molecules are going to be able to get through this hydrophobic core and get into the cell freely. And so this is going to be things like steroids. So think about testosterone, estrogen, which are steroid hormones. Those can cross the cell membrane on their own. Also, small hydrocarbons. Remember that carbon and hydrogen, similar electronegativities, and so when they bond, they're going to form non-polar covalent bonds, meaning they're going to share the electrons equally, and they're also going to be hydrophobic. And so small hydrocarbons can cross the membrane on their own, as well as non-polar small molecules like carbon dioxide and oxygen. Because again, if they're non-polar, they're also going to be hydrophobic, which will allow them to be able to get through this hydrophobic core of the cell membrane. The things that cannot cross are things that are hydrophilic, and that's because hydrophilic molecules cannot interact with this hydrophobic core. So think of hydrophilic like water can't interact with oil, which is like this hydrophobic part. So a good rule of thumb is if something dissolves in water, it can't cross the membrane on its own. And so that includes things like salt, right? If you remember back to salt, salt is sodium chloride, and those are ions. And sodium is a positive ion, chloride is a negative ion. These are hydrophilic. They'll interact with water, and therefore they cannot get through this hydrophobic core, and these ions can't cross the membrane on their own. Another example, glucose, right? Think of sugar, for example. If you took sugar and you dissolved it in your coffee, it would dissolve. And that's because sugars are polar. And if they're polar, they're hydrophilic. And again, things that are hydrophilic cannot cross this hydrophobic interior of the membrane. Amino acids, which are, remember, the building blocks for proteins, those are also polar and therefore cannot cross this hydrophobic core. So the things that cannot cross, hydrophilic molecules, charged ions, or very large molecules. All of those components make it so they cannot cross the membrane on their own and they need help. So we're going to talk now about diffusion. And diffusion is the movement of molecules or ions from a region of higher concentration to a region of lower concentration. And so I want you to imagine for a minute that you have a beaker of water or a glass of water. If you take that glass of water and you put in drops of food coloring, for example, you'll notice that when you put that food coloring in, those drops of dye don't stay as drops forever. That dye is going to start to move and spread out. It's going to go from its high concentration as a drop to the low concentration, which is the water around it, right? Because when you put the drop in, the high concentration is where you put it in. The low concentration is going to be the water around it. And so those dye molecules are going to move from a high concentration to a low concentration. And they'll continue to move and they'll continue to spread out until eventually they're evenly distributed in the water. And at that point, we would say that they've reached equilibrium. Now, when we talk about things going from a high concentration to low, you'll hear the term going down the concentration gradient. And if you think about it, going from high concentration to low, right? So that dye going from its high concentration to low, that's a favorable reaction. That will happen on its own. I don't have to do anything to make those dye molecules spread out. And so when we talk about a concentration gradient, there's two ways. Things can go up a concentration gradient or they can go down. And now think about which one is more favorable. Which one's going to happen spontaneously? Will things roll, let's say, down a hill, or spontaneously will they go up a hill? And you guys probably all know, based on gravity, that by default, without anything happening, things would go down a hill. That's a spontaneous process. Going down a hill doesn't require anything. Going up a hill, though, requires energy. And so diffusion, because we're going from higher to lower, and that's favorable and that happens spontaneously, we say that that's moving these molecules down the concentration gradient. And again, this is a spontaneous process. No energy is required, but it is dependent on the thermal motion of molecules, because remember that molecules have inherent movement. They bounce around. And if you think about what you learned back when you were a kid, if you heat something up, what happens to molecular motion? And if you think about it for a minute, you might recall that if you heat something up, molecules move faster. And so if those molecules are starting to move faster, do you think that diffusion is going to happen faster at warmer temperatures or at cooler temperatures? And so think about that for a minute. Will it happen faster at warmer temperatures or at cooler temperatures? And you might come up with that it's going to happen faster at warmer temperatures, because at warmer temperatures, molecular motion is going to speed up, which means that those molecules are going to move faster and they're going to evenly distribute at a faster rate. And so again, the molecules are going to bounce around and eventually spread out until they reach equilibrium. And so here's a different example of this. Here on the left, we have this beaker, and it has a membrane, and this membrane is permeable to the dye, meaning that these molecules of dye can cross the membrane. And so if we put dye only on one side, these molecules of dye are going to move from their higher concentration on the left to the lower concentration on the right. But notice that the dye molecules don't exclusively go to the right. Some of them by chance will happen to go back to the left. It's just that net movement, meaning it's more going to be towards the right. And those molecules are going to move towards the right until they evenly spread out, at which point we would say that they're at equilibrium. Now, when molecules reach equilibrium, does that mean they stop moving? And the answer is no. Molecules always move even at equilibrium. It's just that those molecules don't have net movement, meaning they're not going one way or the other faster in either direction. The movement to the right equals the movement to the left. And so that's an important concept, is molecules always move even at equilibrium. So water also has thermal motion, and it will also bounce around and work to spread out. And water will also move down the concentration gradient, meaning that it will go from its high concentration, so that's what these brackets represent, that refers to concentration. So it's going to go from its high water concentration to its low water concentration. And so to understand how this works, we need to kind of review and talk about water. And so remember that water, H2O, is an oxygen covalently bound to two hydrogens. And if you remember back to our water lecture, you'll remember that water is polar, meaning that even though oxygen and hydrogen share electrons, they don't share equally. And that has to do again with electronegativity. And if you remember back to that lecture, and we talked about electronegativity, is oxygen or is hydrogen more electronegative? And so remember that we said that the atom that is going to have a greater electronegativity is going to be the one that has its outer shell more full. And so if you remember back to oxygen, oxygen has 6 valence electrons in its second shell of electrons, which means that oxygen only needs 2 more to fill its outer shell. Hydrogen, on the other hand, has 1 electron in its first shell, and it only needs 1 more to fill its outer shell. So hydrogen is only half full, whereas oxygen has 6 out of 8, meaning it's more than half full. And so if you think about oxygen and hydrogen, that then means that oxygen is more electronegative than hydrogen. And what that means is that because it's more electronegative, when they share electrons, they don't share equally. It's like a tug of war. Oxygen wants those electrons more. And so the electrons are going to spend more time around the oxygen and less time around the hydrogen. And so as oxygen pulls harder for those electrons, oxygen gets a partial negative charge because electrons are negative. And if the negatively charged electrons spend more time around oxygen, it's a partial negative charge, which means then that the hydrogens get a partial positive charge. And that's because the electron spends more time with oxygen, which leaves hydrogens as just a proton, and the electron is spending more of the time around oxygen, so it's partially positive. And so what you get is water again is polar. It has a part that's partially negative, which is the oxygen, and a part that's partially positive, which is the hydrogens. Now, when we talked about solutes, remember that a solute is something that dissolves in water. So in this diagram showing it as a sugar, but it just as easily could be something like salt, right? You know that you can dissolve salt in water. So I'm going to show you a diagram using salt as an example because it's a little more simple. And if you remember back to our lecture talking about salt, remember is just simply sodium ions and chloride ions. And sodium ions are positively charged. Chloride ions are negatively charged. And so remember that when we talked about interactions between atoms, opposites attract. And so what's going to happen is that negatively charged chloride ion is going to be attracted to the partial positive hydrogens. And we get these hydration shells. Sodium, on the other hand, has a positive charge and it's going to interact with that partially negative oxygen. And so what happens is the reason that salt dissolves in water is because the water molecules interact with the sodium and the chloride and they separate them from each other. So if on the left you have a lower solute concentration and on the right you have a higher solute concentration, that means that on the right side, where we have more sugar, we have less free water. And what I mean by this is, in this case, this membrane is selectively permeable. And what that means is that some things can cross the membrane, others cannot. And in this case, our solute is not able to get across the membrane. Only the water can move. And so we can't say that the solute is going to move from the right to the left because it can't cross the membrane. So what we need to focus on is which direction the water will move. And so to think about which way the water will move, wherever we have a higher solute concentration, wherever that solute's present, water is going to be attached to it. And if you remember, the solute can't cross the membrane. And so on this side where we have more solutes, we have less free water because the water's not free to move. It's bound to the solute. On this left side here, we have a lower concentration of solutes, but we have more free water. More water that's not bound to a solute that's free to move. And so we have more free water here, less free water here. And remember that things are going to go always from high concentration to low. So it's going to go from its high concentration of water here to low concentration of water here. And water is going to move to the right and it's going to start to fill up the tube on this side. And so again, just like all other molecules, water is also going to move from a high concentration to low. And when you're looking for which way water moves, it's always going to be from the low solute concentration, which is where you're going to have more free water, to the high solute concentration, where you have less free water. So either way is fine to remember it if you want to remember that it moves from low solute to high solute. But for me, I would rather remember that things always go from high to low. And so I always like to remember that it's going to move from here, where it has more free water, or a higher water concentration, to this side where it has a lower water concentration. And so this is going to be referred to as osmosis. Okay, and osmosis is the diffusion of water. Now, tonicity is the ability of a solution to cause a cell to gain or to lose water. And you always need to compare two solutions because a cell is only going to gain or lose water depending on solute concentration. And in order for this to work, it must be separated by a water permeable membrane, meaning that water can move. because how can a cell gain or lose water if water can't move? And you have to be really careful when you start talking about tonicity, because you need to pay attention to which of the two solutions you're referring to. And I'll talk about that in just a minute. So the first one that we'll talk about is a hypertonic solution. And if you think of a kid that's hyperactive, do they have more energy or less energy? And you'll probably recall that if a kid is hyperactive, they have more energy. So the word hyper refers to more. The hypertonic solution is the one that has a higher solute concentration. And so notice that in this case, here in white, this is our cell, and our cell is placed in a solution, and that solution has more solutes outside. And so we would say that the solution outside is hypertonic relative to the cell, because again, hyper means more. So this solution is hypertonic relative to the cell. And remember that anywhere that solute is, water is going to be associated with it. So all these little red dots here are referring to water. And notice that if there's more solutes outside, that means that this outside solution has less free water. Notice that there's only, in this case, one water molecule that's free to move. If we look inside the cell, which has a lower solute concentration, it's going to have more free water because the solute concentration is lower and there's more water molecules that are free to move. So inside the cell, we have more free water. And so think about it for a minute. Which way is the water going to move? Is it going to go into the cell or is it going to go out of the cell? And so think about it. So where is the water going to go? And if you think about it, it's going to go from its high concentration inside the cell to the low concentration outside. What you're going to see is that water is going to move out of the cell. Now, we're going to talk about for each of these solutions what you're going to see in both an animal cell and in a plant cell. And remember that a difference between plant and animal cells is that plant cells have a rigid cell wall. Animal cells do not. And so this is going to affect the way that water moves in a plant cell versus an animal cell. So in an animal cell, when the water moves out from a hypertonic solution, so as the water goes out, animal cell is going to shrivel up. For the plant cell, the water's still going to go out, but the whole cell is not going to shrivel up because it has that rigid cell wall. Instead, you're going to see something called plasmolysis. And what that means is that as the water goes out, the cell membrane is going to shrink in away from the cell wall. So it's like the membrane kind of collapses in, but the cell wall stays out here. And so in plant cells, this is referred to as plasmolysis. And you're actually going to visualize this in lab this week. Now, let's look at what's going to happen if you place a cell in a hypotonic solution. And hypo is less. So remember that if hyper is more, hypo is the opposite, it's less. So in this case, it's the solution with a lower solute concentration. So in this case, the solution outside is hypotonic relative to the cell because it has a lower solute concentration compared to the cell. And so when we look at water concentration, remember that if we have more solutes inside, less solutes outside, that means that we have more free water outside. And so think about it. Which way is the water going to move if you put a cell in a hypotonic solution? And so if you think about it, water is going to go from its high concentration in the solution to the low concentration inside the cell. And the water in this case is going to move into the cell. And the way that I remember that is if you put a cell in a hypotonic solution, water is going to go in, the cell is going to swell, and it's going to become a big O. So hypo, cell is going to become a big O. Okay, and so water is going to go in, cell is going to swell. And so that's going to look different if you talk about an animal cell versus a plant cell. In an animal cell, if you put it in a hypotonic solution, the water's going to go in, the cell's going to swell, there's no cell wall to restrain how much water goes in, and the cell's going to lyse and it's going to burst open. Think about for a minute if you've ever been dehydrated and you had to go to the hospital to get an IV. You probably know that if you go to the hospital, they're not going to put pure water into your IV. They're going to put a saline solution. And that's because they don't want to create a hypotonic solution outside your cells so that all the water is going to rush in and your cells are going to burst or lyse open. And you can actually die from water intoxication. It's something called hyponatremia. And it happens sometimes in marathon runners when they sweat too much. If they drink just pure water, you create a hypotonic environment outside the cells. Too much water goes in and your cells would start to lyse. And so again, this is why in a hospital, if you're dehydrated, they're not going to put pure water in there. They're going to put a saline solution that has some solutes so that you don't have too much water rushing into your cell. If you think about a plant cell, however, if you water a plant, you don't use salt water or saline to water your plant. You just use tap water where the solute concentration is low. You are putting your plant in a hypotonic solution. And in a plant cell, that's actually a favorable reaction because as the water goes in, it creates some pressure inside that cell, and that makes the cell become turgid. It creates what's called turgor pressure. And that pressure inside the cell from the inside pushing on the cell wall, that pressure is going to make the cell very rigid, very firm, and that's what allows the plant to stand upright. It needs that turgor pressure. So in a plant, you want to put the plant cell in a hypotonic solution because you want the water to go in to create that pressure. But again, animal cell, not good to put it in a hypotonic solution because the water is going to go in, no cell wall to restrain it, and the water is going to cause the cell to go pop and burst open. That's called lysis. Now, back to the slide about tonicity, and I said about always make sure to pay attention to which solution you're which solution you're talking about. So notice in this case, we can say that the solution is hypotonic, but the cell we could also call hypertonic. So we could say that the cell is hypertonic relative to the solution, right? It has more solutes relative to the solution. So again, always be careful when you're using the terms hypertonic and hypotonic. Which one are you referring to? Are you talking about the solution or are you talking about inside the cell? And so in this scenario, the solution is hypotonic relative to the cell, or you could say the cell is hypertonic relative to the solution. And the last scenario is if you put a cell in an isotonic solution. Iso refers to same. And this is where you have an equal solute concentration on both sides of the membrane. So notice 1, 2, 3, 1, 2, 3. Solute concentration is the same, which means that the water concentration is also the same. Notice 1, 2 free water molecules, 1, 2. And so what that means is you have an equal concentration of water on either side of the membrane. And so think about that for a minute. Does that mean that the water won't move? And if you think about it, right, you might recall that molecules always move even at equilibrium. And so it's not that you don't get any movement, you just don't get net movement. Water is going to go into the cell and out of the cell at the same rate. And so the water is equally going to move in both directions. So if you put an animal cell in an isotonic solution, water is going to go in and out at the same rate. That's going to be a normal animal cell. Again, you want your cells to be in an isotonic solution. For a plant cell, however, that's not a good thing. Again, you want that pressure inside the cell in order to make the cell firm so that the plant stands up. if you were to water your plant with an isotonic solution, where the water is going into and out of the cell is going to be flaccid. And if it's flaccid, it's not firm, and your plant's going to wilt. And so, best scenario for an animal cell, isotonic solution. Best scenario for a plant cell would be to actually put it in a hypotonic solution so that the water goes in. And so I have a class paper for you. So if you're stranded on an island, should you drink the ocean water to quench your thirst? Why or why not? So when you're ready, go ahead and pause this and think about your answer and write down what you would have answered had you been in class. And then when you're ready, go ahead and turn the video back on and listen to the answer. So go ahead, pause it. Okay, so let's go over the answer. So if you're stranded on an island, should you drink the ocean water? The answer is no, because if you drink the ocean water, which is salt water, right? Think about what happens. If you drink ocean water, you're creating a hypertonic solution outside your cells, which means there's more free water in your cells, and the water is going to go out of the cells, and that's going to cause your cells actually to become more dehydrated. And so if this continued to happen, this would lead to dehydration and in extreme cases, death. Okay, and so you would not want to drink pure ocean water because, again, drinking salt water would cause you to become more dehydrated. And then this last one, here's a series of questions, and the answers will be posted on Blackboard for you to use to study. So I want you to work on these, and then after you've worked on these, you can check your answers with those that are posted on Blackboard. And so this is going to conclude part 1 of the video. And when you're ready, you can go on to part 2. And we're going to start by talking about transport of molecules or ions across the membranes. So how do things actually get into or out of the cell? So the first type of transport is referred to as passive transport. And it's passive because no energy is required. This is the use for the transport of molecules or ions down the concentration gradient. And remember that means moving things from a high concentration to low. And going from high to low, remember, is down the gradient because that's favorable, right? If I put a ball on the edge of a hill, it's spontaneously going to go down. And same thing for molecules going from a high concentration to low, that's a favorable reaction. Right? Remember our beaker with water? If I put dye into that beaker, it's going to move from its high concentration as a drop to the low concentration in the solution surrounding it. And so again, passive transport is used to transport molecules or ions down the concentration gradient, which is a favorable reaction. And there are 2 main types of passive transport. The first is going to be simple diffusion. And in simple diffusion, no transport protein is necessary. This is used for things that can cross the plasma membrane on their own. And remember that we said that the things that can cross the membrane on their own without the help of a transporter are going to be things that are small, hydrophobic, and nonpolar. And so things that are small, hydrophobic, and nonpolar can get through that interior of the plasma membrane, which is also hydrophobic. So things like steroid hormones, testosterone, estrogen, those are made of primarily hydrocarbons, and hydrocarbons are nonpolar and therefore hydrophobic. Molecular oxygen, so oxygen gas, carbon dioxide, those are both nonpolar. They can cross the membrane all on their own. The only requirement for passive or for simple diffusion is that it's going to move from a high concentration to low. And so notice that in the image that you see here, it's going to move from its high concentration to low. So the only thing that's required is the concentration gradient. Now, in contrast to simple diffusion, we have what's referred to as facilitated diffusion. And this is basically that the diffusion is facilitated by these transport proteins. So this is used for things that cannot cross the membrane on their own. They still will go down the gradient, meaning they'll go from the high concentration to low, but these molecules can't get through the hydrophobic core of the cell membrane. And so what's needed is this transport protein. And this transport protein, like for example this one here, that's a channel protein, and it basically provides a hydrophilic corridor, meaning that the amino acids from this protein that are in the middle are also hydrophilic, which allow these hydrophilic molecules to be able to cross the membrane. And so this is going to be used for ions and polar molecules. An example of this, aquaporins. If you think of aqua, you think of water. Porins are pores. Aquaporins are channels that allow water to cross into or out of the cell. There are also what are called carrier proteins. And these are transmembrane proteins that not only provide a corridor for things to cross, but they actually change their conformation, which is their shape, upon binding to the solute. So on one side, it'll open up, the solute will come in and bind, the transport protein will change its shape, and now open up to the inside and release that solute into the cell, for example. Again, these are going to go from high concentration to low because they're passive transport. They don't require energy. This is in contrast to active transport. And in active transport, it requires both a transport protein to help move that molecular ion, plus it requires energy. And that energy that's required is usually in the form of ATP. And this type of transport is used to transport molecules or ions up the concentration gradient. Up, remember, if you were to push something up a hill, up requires energy. So you would have to put in energy to get a ball, say, up a hill. Same thing if you're going to transport things up the gradient. If you're transporting things up the gradient, it requires energy. And this is used to transport things from a low concentration to a high. If you think about that beaker that has water and has a drop of dye in it, that dye, remember, is naturally going to go from its high concentration as a drop to the low concentration around it. And if you think about it, is that dye ever going to go from a low concentration to high? Meaning, is it ever going to go from where it's spread out and all of a sudden spontaneously concentrate itself back into a drop? And the answer is no. Things will not spontaneously go from a low concentration to a high concentration. And so that requires energy, which is why we call it active transport. You actively have to do something to move these molecules or ions up the gradient. Some examples of this, ion pumps. For example, here you're seeing a proton pump. And a proton pump, like you can imagine, pumps protons. Protons are simply hydrogen ions. Remember that all hydrogen is. A normal hydrogen atom is one proton, one electron. When we talk about a hydrogen ion, in a hydrogen ion, it gives up its electron. And what you're left with is H+. It has a positive charge, because all that's left is a proton. So that's why we call H+ protons. And in the proton pump, we're going to pump hydrogens against the gradient. So from a low concentration to a high, and because that's an unfavorable reaction, it requires energy in the form of ATP. Now, this type of pump is what we call electrogenic. And what that means is that it generates a charge difference across the membrane, what we call a voltage. So as we pump hydrogens into the extracellular fluid, which is the fluid outside the cell, you're creating an environment where the extracellular fluid is positively charged relative to the inside of the cell. And so that's creating a voltage difference. It's a difference of charge across the membrane. Another example that doesn't have a diagram here, but is still important, is the sodium-potassium pump. And sodium-potassium pumps are used for neurons. When a neuron normally does an action potential, okay, meaning that the neuron fires, that is caused by a change in the voltage across the membrane. And in order to turn off that neuron again, sodium and potassium pumps need to go against the gradient to reset that voltage so that the neuron can fire again. And so again, these are going to be electrogenic. They generate a voltage or a charge difference across the membrane. Co-transporters requires two transmembrane proteins. One is going to pump a molecule up the concentration gradient, so meaning it's going to go from a low concentration to high, and one that's going to let the molecule flow back down the gradient. And so the downhill movement is coupled to the uphill movement of another molecule. And an example of this is the sucrose proton transporter. And so if we look at this cotransporter, here it's coupled with a proton pump, and a proton pump is going to pump hydrogens against the gradient. So again, moving it from its low concentration to its high concentration out here. Now, as hydrogens go down the gradient, going from high to low, that energy that it gives off by going down the gradient is enough to help sucrose get into the cell. And so again, this is referred to as cotransport. It is active though, because it requires either ATP or some other favorable reaction to power the movement against the gradient. So now we're going to talk about bulk transport across the membrane. So how do we get large, bulky molecules either into the cell or out? And we're going to talk about exocytosis and endocytosis. Exocytosis, okay, exo is referring to exit. This is how the cell gets large things out of the cell. And the way that this works is that these lipid vesicles are made in the cell, and eventually those vesicles fuse with the plasma membrane and release the contents to outside the cell. And if you remember in our lecture about cell structures, we talked about the protein production pathway. And in the protein production pathway, proteins go from the rough endoplasmic reticulum, they bud off in a vesicle. That vesicle takes the protein, it fuses with the Golgi apparatus. And the Golgi apparatus is going to modify, it's going to sort the proteins, and it's going to ship them. And that protein's going to leave in a vesicle, and the vesicle moves along, it fuses with the cell membrane, and it releases those secreted proteins out of the cell. And so this is how a cell secretes materials, like, for example, proteins, again, for our protein production pathway. Hormones, so insulin, for example. Insulin is made in pancreatic beta cells. So in your pancreas, you have a special group of cells that's responsible for producing insulin. And when you eat, your body recognizes that sugar is available. So when you guys eat, your blood sugar begins to rise. And as your blood sugar goes up, your body recognizes that there's sugar available. And your pancreatic beta cells produce insulin, and they release insulin into the bloodstream. Insulin travels throughout your body, and it serves as the signal to tell your cells to take up glucose. Because all the cells in your body require glucose for energy to make ATP, so for cellular respiration, for example. And so insulin is that signal that tells your cells that glucose is available. And so cells in the pancreas need to release that insulin so that that hormone can go through your bloodstream and circulate and tell all the cells in your body to take up glucose. This is also how neurotransmitters get released. And so the way that two neurons communicate with each other is that one neuron that's already fired is going to release a neurotransmitter to an adjacent neuron. When that adjacent neuron receives that neurotransmitter, now you get an action potential. You get a neuron to fire for the adjacent cell. And so neurotransmitters are how neurons communicate with one another. This is also how the cell membrane grows. Because if you think about it, these vesicles that are made here are basically components of the cell membrane. And so as these vesicles fuse with the cell membrane, you're going to increase the surface area of the cell membrane, and you're going to increase the cell membrane. Now, in contrast to exocytosis, which is how we get things out of the cell, endo refers to in, getting things into the cell. And so endocytosis is how we get large molecules or particles into the cell. One of the types of endocytosis is something referred to as phagocytosis. And in phagocytosis, you can think of this like cellular eating. And what the cell does is it sends out these extensions, and these extensions are referred to as pseudopodia. And if you remember back to our lecture on cell organelles, remember that we talked about cytoskeletal elements, and specifically we talked about actin. And actin is the cytoskeletal element that's responsible for producing pseudopodia. If you remember into our video of the white blood cell chasing the bacteria, when that white blood cell came to the bacteria, it sent out those projections and engulfed and took that bacteria into the cell. That's phagocytosis. Okay, and so the cell is going to send out these pseudopodia. It's going to fuse the pseudopodia together and engulf that food and other particles into the cell into a food vacuole. And then that food vacuole can get broken down and take in the food. Or if it's not food, if it's, let's say, bacteria, okay, the cell is going to engulf that bacteria. The white blood cell will engulf the bacteria. The bacteria, remember, would fuse with the lysosome, and the lysosome would then break down that foreign bacteria. And so this is used to ingest larger particles, things like bacteria, viruses, fungi, lots of food molecules, for example. All of these things would use phagocytosis. This is in contrast. Another type of endocytosis is referred to as pinocytosis. Pinocytosis is pinching in. So the cell in this case does not send out extensions. It simply pinches in, and it creates kind of this pocket. And this little pocket is going to take in any of the liquid that's outside here. And it's a very non-specific type of endocytosis, meaning whatever's out here is going to be incorporated into that vesicle. And you can think of this kind of as cellular drinking, because again, it's not going out, it's not taking in food. All it's doing is the membrane is pinching in. Any fluid that's outside is going to become incorporated into this vesicle. And that's how the cell's going to get in some of the solutes that it uses. And the last type of endocytosis is something called receptor-mediated endocytosis. So receptors are proteins, and they have a binding site for a type of molecule referred to as a ligand. And receptors are very, very specific. So if we look at this receptor, it has a ligand that would bind to it, and a cell would recognize when the ligand bound to the receptor, and when it did, it would form this coated pit, and it would take in large amounts of this ligand in a very specific manner, because again, these receptors are specific for certain ligands. And so this allows the cells to take in very specific ligands, very specific types of molecules. Example of this, LDL. So if you've ever had your cholesterol checked, you've probably heard a little bit about LDL. LDL stands for low density lipoproteins. And these low density lipoproteins, these are these cholesterol carriers. And remember that cholesterol is important for the cell. Again, even though we think of cholesterol as being a bad thing, cholesterol is needed. It's used to make steroid hormones like testosterone, estrogen. Cholesterol is also used in the cell membrane in order to regulate the correct fluidity of the membrane. And so one of the ways that cholesterol gets into the cells is that these LDLs, these cholesterol carriers, LDLs are going to carry the cholesterol, and LDLs bind to these LDL receptors. These receptors that bind to LDL. And when they bind, it's going to take the LDL into the cell and out of the bloodstream, and that's going to allow cholesterol to get into the cell. And so LDLs compared to what we call HDLs, LDLs take cholesterol towards the heart. HDLs, so high density lipoproteins, takes cholesterol away from the heart. And so typically you want to have a lower amount of LDL because you don't want to take a bunch of cholesterol, a bunch of lipids back to the heart. And you want a higher number, not too high, but a relatively higher number for your HDLs, which are the ones that are going to take the cholesterol away from the heart. And again, this is referred to as receptor-mediated endocytosis. It allows the cell to take in large amounts of a specific type of ligand. So, class paper for you. Familial hypercholestemia is a genetic disorder in which there are defective LDL receptors. What is the likely outcome? So think about it for a minute. Okay, what would happen if somebody was born with defective LDL receptors? What would be the result? So I want you to think about it for a minute, and then you're going to pause it while you're thinking. And when you're ready, go ahead and push play. Okay, so if somebody is born with defective LDL receptors, what that means is that if their receptors don't work properly, they're not going to be able to take in LDL. And remember, LDL are the cholesterol carrying molecules. And if they can't take in LDL, what that means is that that LDL, and therefore that cholesterol, is still present in the bloodstream. It's not going to come out of the bloodstream. And what happens as a result is people who have this condition have genetically high cholesterol because their cells cannot take in the LDL to get it out of the bloodstream. And so because it's not taken up by the cell, it's going to lead to an increase in LDL and cholesterol in the blood. And if left untreated, heart disease can occur. So if your parents have high cholesterol, it's always a good idea to get checked because it could be a consequence of not necessarily diet, but it could have to do with just simply genetics. I'm one of those people. I've had high cholesterol ever since I was a teenager. No matter how much I exercised, no matter how well I ate, I always had high cholesterol. And again, that's because I have defective LDL receptors. So it's not that I'm getting too much cholesterol in my diet. It's simply because my cells can't take in LDL, and therefore the cholesterol stays in my blood. And so if you happen to have high cholesterol, you want to go and find that out and get on medication to lower your cholesterol. Now, currently the medication that they prescribe for high cholesterol doesn't really fix the defective LDL receptors. There's not really a good way at the moment to make functional LDL receptors. Instead, what those drugs typically target, if you take like a statin, which is like Lipitor, lovastatin, there's a bunch of them. Basically, those drugs don't target your LDL receptors. Instead, they actually target the liver to stop making cholesterol. Because if you don't make as much cholesterol, that will help to keep your cholesterol down. And so, just something to think about.